Good day, dear ladies and gentlemen. This is our collaborative talk on space biology with Galina Nechitailo. This lecture is our view on the problem of biological support for future space missions. It is clear now that spaceflight factors influence on biological objects. How can we make space garden in such case? What experience do we have today after almost 60 years of human spaceflight? This is a large problem and cannot be overviewed in one short talk. I would focus on Energia results, what was hidden for a long time due to different reasons, while other studies can be found in the literature. This is one reason of this focus. Another reason is that these studies were directed to make a space garden as biological support for future long-term space missions, to Mars, for example. And I don't know other massive biological studies in space flights that was so focused on space gardening. Long space journey with a crew required a life support system to provide food, air, and water for crew members or humans. If journey is around the Earth or Moon, all the stuff can be provided from Earth, as it is working now for International Space Station. But if Mars or beyond mission, the provision from Earth would be unrealistic. We would need space garden for food production and for air and water regeneration. Such space garden must be successful for years, stable and productive, otherwise death of astronauts. There are two possibilities for successful space garden: to get harvest in space and regrow it again and again from seeds, or to take enough seeds to get harvest from Earth seeds every time. First way is more effective and make space journey unlimited, but this way is more complicated. Second way is viewed more robust, but amount of taken seeds limits the time of space mission. Both ways were considered from beginning of space era. For example, Tsiolkovsky considered space garden as a solution for long-term space missions. He wrote that humans and plants will be affected by unusual space factors such as microgravity. Another fact of space flights was discovered later, during stratospheric flights in 1930s about. This is cosmic rays. At that time, first small animals, insects and plants were sent to stratosphere on balloons. Later, the investigations were continued with sound rockets up to 500 km altitude. Real spaceflight investigations were started with orbital flights. First satellites were used for animal experiments. The first organism sent in its orbit on a board of second satellite was Laika, a dog. It survived. Third, fourth, and fifth satellites carried out a biological payload with organisms. Then a number of biological experiments were installed on manned and unmanned satellites and spaceships. Even first manned spaceflight of Yuri Gagarin carried out the Drosophila experiment. It was found that situation with living organisms in spaceflight is complicated, and it was clear that there is a strong influence of space environment on humans, animals, seeds, microorganisms, on everything what was grown on Earth and what was adapted to grow and develop on Earth. Space is different. However, short flights didn't give a real possibility to investigate the influence of spaceflight factors on biological systems. It needed a long-term flight experiments, as minimum as during a whole life cycle. New possibilities were opened with first long-time space missions. First vegetation of plants in space was done during 18 days orbital flight of Soyuz 9 spaceship. At this time in 1970, a group of biologists in Energia company was hired. Their task was to coordinate biological missions, design and install biological flight equipment, carry out biological experiments during space flight, and conduct after-flight investigations. As a goal was a space garden. This activity was a part of the space program for a long time. Then the systematic biological studies on Salyut and Mir stations became available. In this flight on Soyuz 9, the experiments with growing chlorella and seeds were installed. The first potato was growing in space. Next flight, Soyuz 10, was a short mission, but the first tulips was blossoming in space flight. More possibilities appeared when first orbital station Salyut 1 was launched. Massive space biology program has been done during this flight. 23 days in station, in space, in orbit. First experiments was growing plants. First vegetation system on the board. First green harvest grown in space. It was a great success. However, this flight was a tragedy. Cosmonauts were died at descent. The venting valve for spaceship was faulty and open at high altitude. Next flights were short before spaceships were sufficiently tested. However, the biological experiments continued. Even just beginning of growing phase of the plants was an interest. First self-sufficient ecological system was tested in Soyuz 13 in 1973. Urobacteria produced hydrogen. Hydrogen oxidizing bacteria eat hydrogen. It was a cycle. The system works stable on Earth and in space. In this flight, Chlamydomonadas proteus was growing in IFS-1 device and ROST-1 device. In 1975, the biological equipment was installed in Salyut 4 space station. A massive pool of biological experiments in space has been started. For example, the first experiment with fish in orbit was done at that time. You can see the Oasis 1M system for cultivation of plants in right corner. Such devices were permanently installed in the station on ground and tested before the launch. In flight, in orbit, only sets with substrate and seeds were changed during each mission. The devices provided fixed position of the plants in artificial substrate, regular watering, aeration, and illumination of the plants. At the same time, the devices provide safety of the biological experiments for crew and for other devices on the station. Unique and suitable equipment was designed, tested, and used such as Svetablock, Oasis, Phyton, Vazon for Salyut and Mir station. In this flight, for the first time, green onion was grown in space. First green onion were eaten by astronauts. And astronauts survived. First food grown and eaten in space flight. The international crew, as the result of Russian-American cooperation, also did some short biological experiments. The microgravity was always a problem for the high plants. These first experiments with gravistat were done to provide the artificial gravity on the board of spacecraft. You can see section of first cone roots grown in biogravistat in the right bottom corner. Fungi were growing in Salyut 5 station. The different regimes of growing were investigated. Fungi showed different morphology. That was first fungi grown in space flight. Next station, Salyut 6, carried out a lot of biological experiments. For example, first experiments with medusa set were done. You can see it in right corner. This experiment is related to abiogenic synthesis outside of station in free space. The first experiments on origin of life. The cassette was exposed in free space for years. From station Salyut 6, the biological experiments were installed permanently and managed by permanent crew. First experiments with tissue culture were done in space flight. For these experiments, a line of space flight thermostats was developed, designed. You can see it in right corner. Here you see cosmonauts Kovalyonok and Ivanchenkov with phyton device with onion grown in space flight. The photo has been done during communication with the flight center with biologists. The phyton device was designed in such a way that growing plant doesn't contact with atmosphere of the cabin. Seeds were outshoot from seeds gone into the substrate, where they started to germinate. A number of international crews were involved in these biological experiments. For example, here is Polish cosmonaut. Here is a German cosmonaut. These were short visiting missions. You see a list of biological experiments carried out by visiting crews. Permanent crews were loaded with a long biological program as usual. For example, for the first time, electrostimulation of the plants in space flight was used. Artificial electrical field was applied during seed germination, root initiation, and plant growing. The device was compatible with electronic devices of the space station, and it was safe for crew and station operation. You can see it at the bottom, this device. For the first time, eggs were developed in space flight, and a lot of other interesting results with biogravity to replace the gravity in other plants. In this flight, there were a wide number of experiments too. In particular, there were experiments to minimize the radiation damage effect with anti-mutagen agents. This is important to store seeds for a long time in space flight. Cosmic radiation causes radiation damages in seeds. Anti-mutagen agents successfully reduced this effect. Also, for the first time, Arabidopsis was blossoming in space flight. It was grown from seeds in phyton system. This flight was full with biological experiments. Here is a long list. Probably the most interesting are weed growing in our system. You can see it in the bottom left image. Electrostimulation of pea growing, biorhythm of Drosophila, and experiment in magnetogravistat. The last experiment is if a magnetic field can orient seedlings instead of gravity. Flags on board of Salyut and Mir station were growing 3-4 days in magnetic field gradient. The seeds grow along the field gradient direction. You can see seedling orientation in bottom stage, different versions. Next step is Salyut 7 space station. Cosmonauts Berezovoi and Lebedev, a permanent crew, have done a wide number of biological experiments. Electrostimulation of pea plants, leaded gravistat, microorganisms kinetic of growing, proteus growing kinetic, seeds exposure, orchids growing, abiogenic synthesis in free space. Point out, at the time, the microorganisms resistance to antibiotics was tested during the microgravity and found the increase of resistivity in space flight. Visiting expeditions helped to carry out the experiments. For example, crew Soyuz T6 took part in a number of biological experiments, particularly in orchids experiments. This visiting expedition came in right time. The Arabidopsis gave first harvest in phyton system. It was the first experiment with growing plant from seeds in space, which gave seeds-to-seeds experiment. The possibility to get a harvest in space flight from seeds was shown for the first time. A path to long-term flights with biological life support system is open. Next permanent crew, Lyakov and Alexandrov, worked 149 days in station. They continue biological experiments with cultivation of plants and seeds. Next permanent crew also continue biological experiments with plants cultivation, anti-mutagen agents, seeds, and abiogenic synthesis. Experiments repeat for statistics, for new agents, for new components, for new plants. Soyuz T-13 was a heavy flight. Cosmonauts repaired the station, which was frozen due to power problems. They restarted biological systems and long-term experiments again, because previous plants were frozen and dead. But when the station has come to work back, they did a large number of biological experiments. In particular, cotton was growing in space for the first time. The cotton grown in space had almost double length of fibers than the longest cotton fibers known on Earth. They started experiments with experimental cuvette with artificial substrates. It was important for optimization of plant cultivation. They started experiments on polymerization of acrylamide gel. The gel synthesized in space was used for protein separation in electrophoresis. The separation with space gel gave much higher resolution than usual technology with ground polymerized gel. Last expedition to Salyut 7 station was shortened due to health problem of one cosmonaut, while cosmonauts carried out a number of successful biological experiments. New period of space biology has been started with Mir station. From first expedition, each crew carried out biological experiments on station. By volume of data, about half of experimental results were received from Mir station. These are genetic studies, morphological studies, harvesting first wheat, first birds from eggs, snails, concerts, first radish harvest, first cabbage harvest, first kidney beans, first tree in space. Tissue culture experiments gave increased biological activity of plants, first potato harvest, first result is long-term seeds exposure to space, 6 years, and got harvest from them. Mir station expeditions were more open for international scientific society, for collaboration. All these results can be found in the literature. These are main stages of plant cultivation achievements in space stations. As you see, it takes a long time to develop new technologies, methods, equipment, but also it shows that harvesting in space is possible. This is the main result of these studies. In design of biological life support systems and space greenhouses, green gardens, these factors must be taken into account. At first, microgravity, then space radiations of different nature and energy, absence of Earth natural electrical and magnetic fields, and presence of local artificial electrical and magnetic field of equipment of space station and spaceship, different light cycling and spectrum. So these are critical factors which change the biology of plants, plant genetics, morphology, growing patterns, biochemistry, and so on. Main results of this study can be formulated here. You can read it carefully if you put pause in the video. It was long-term systematic investigations. It was a unique situation when biologists were involved in design and development of space stations, planning space missions, So we can establish a space garden from seeds taken from Earth for many eatable plants. For long-term space flight, the seeds must be treated with anti-mutagen agents. The harvest in space flight cannot be expected as big as on Earth. Productivity of the plants in space will be lower than on Earth. Content of biological active substances such as proteins, lipids, sugars, and others would be different. Cosmonauts ate it, but it was not their main food. More investigations are required where such products grown in space can be main dish for human. Another way harvesting in space flight and regrow it again requires more efforts. For today we are successful with harvesting of wheat, radish, cabbage, potato and some others in space flights. Eatable plants. These plants can be reproduced in space garden. However, these plants require more space experiments with multiple generations in space, establishing specific environment to get harvest. Other eatable plants are waiting for space experiments. This is where we are today. Thank you.